Okay, I believe we can start. Good morning once again, and welcome to today's lecture. Today we'll be looking at gene expression and protein synthesis. And in session 1, we'll delve into proteins, amino acids, and the relationships between genes and polypeptides. So by the close of this lesson, you should be able to explain relationship between proteins and amino acids and understand and explain chemical structures of amino acids. So without wasting much time, let's first move on to what proteins and amino acids are. So basically, proteins are, food additives that are molecules responsible for catalyzing most intracellular chemical reactions. They are basically enzymes in living organisms, and they are responsible for regulating gene expression and detect many features of the structures of cells, tissues, and many structural approaches in viruses. So basically, a protein is composed of one or more chains of amino acids. And there are 20 different amino acids commonly found in polypeptides that can be joined in any number and in any order. So that tells you how numerous proteins and polypeptides are with regards to the amino acids. We have 20 different amino acids that are found in polypeptides. Polypeptides are very giant molecular protein molecules that are formed from these 20 different amino acids. So because of the number of amino acids in a polypeptide, usually ranges from 100 to 1,000. That is quite a huge number of the different molecules that can be formed from the 20 amino acids. So that means that you could be counting from 2,000 to 200,000. So each amino acid contains, what it entails is that it contains a carbon atom, which is referred to as the alpha carbon. Alpha carbon. So it is from the alpha that the others join, to which one is attached to the carboxyl group, COH, and one amino group. So that informs an amino acid, and a side chain commonly called an R group. An R group could be any other group that is attached to the amino acid. So the general structure of an amino acid is clearly labeled, displayed here in this figure. You have the alpha, the first carbon atom, or the alpha carbon, and the carboxyl group, COH, carbon being attached to hydroxyl, OH, and oxygen, double bond. Then the NH2, that is the amino group that informs the structure of an amino acid. So the R groups are generally chains or rings of carbon atoms bearing various chemical groupings. So the simplest chains are those of glycine and of alanine. So they are CH3. CH3. These are the common R groups that one can come across. Then again, the polypeptide chains are formed when the carboxyl group, and by the carboxyl group, referring to the COH of each amino acid, comes and join with the amino group of the next amino acid in line. So they form bonds. That is why it's referred to as a polypeptide. There are many, numerous of them. hundreds of thousands of them, resulting in a chemical bond, an ordinary covalent bond, referred to as what? A peptide bond. So the basic unit of the protein is a polypeptide chain. The basic unit of a protein is a polypeptide chain. We have a peptide chain that is one amino group, but it comes together collectively. That is why it forms that giant molecular structure referred to as a polypeptide, in which alpha carbon atoms alternate with the peptide groups to form a backbone. The phosphate backbone that it forms, which has an ordered array of side chains. So you could have a lot of other chemical groupings adding up to the backbone. So the two ends of every polypeptide molecule are distinct. One end has what? Amino group, that is NH2, and it's called the amino terminus. Terminus means it is terminating. Remember, in the other lectures, we looked at the 5' and the 3' terminus. The other end has a free carboxyl group, and it's called carboxyl terminus. So what informs the formation of polypeptides? What informs formation of polypeptides? What goes into the formation of that giant molecular protein structure? Polypeptides are synthesized by adding successive amino acids to the carboxyl end of the chain, of the growing chain. So you have one carboxyl end in a growing chain, and you add another. To the R group. Okay, then it keeps on adding. Conventionally, the amino acids of polypeptide chains are numbered, starting at the amino terminus. So, for instance, you could have like naming an organic compound. The number of the longest carbon chain is how you go about naming the amino acid. So conventionally, the amino acid of a polypeptide chain are numbered starting at the amino terminus. So when you have the NH2, that is where you start. Most polypeptide chains are highly folded. A variety of three-dimensional shapes have been observed. So the manner of folding is predominantly determined by the sequence of amino acids, in particular by non-covalent interactions between the side chains. Hence, each polypeptide chain tends to fold into a unique three-dimensional shape as it is being synthesized. So in some cases, protein folding is assisted by interactions with other proteins in the cell called chaperones. So before you would get your protein, you realize that it is as a result of what? The breaking up of the DNA structure, releasing one strand into the cytoplasm to merge with other ribosomes to form your proteins. So basically, that is what occurs, the formation of peptides. So generally speaking, the molecule fold so that the amino acids with charged side chains tend to be on the surface of the protein, and those with uncharged side chains tend to be what? Internal. So you have codons, you have anticodons. The heterogeneous bases will look up for its complementary base pairings, and that is what informs the formation of those polypeptides. So you would have a lot of some of these terminals in the cytoplasm. So specific folded configurations also result in hydrogen bonding between the peptide groups. And as we've been told earlier, it's the hydrogen bonds that hold the nth molecular structure together, the DNA structure. It's the DNA and the hydrogen bonds that hold them together. And when they are unzipping, a DNA polymerase brings about what? The process of unzipping. So two fundamental polypeptide structures are the alpha helix and the beta sheet. We'll be looking at that in subsequent figures. The alpha helix, just like the alpha carbon, represented a coiled ribbon, is formed by interactions between neighboring amino acids. So it isn't just one polypeptide occurring in the cytoplasm. There are numerous of them. You've been told that we have 20 different types of what amino acids occurring, and the numbers are in the hundreds to 1,000. So you can imagine the numbers are enormous. So the interactions are between neighboring amino acids that exist, that twist the backbone into a right handle helix in which the amine, in which in each of the peptide group is hydrogen bonded with the carboxyl in the peptide group located for amino acids further along the helix. So in contrast, the beta sheet, represented as a parallel flat ribbon, is formed by interactions between amino acid in distant parts of the polypeptide chain. So the backbones, that is the phosphate backbone of the polypeptide chains, are held flat and rigid and forms the what? The alpha sheet. So there is a backbone, and you have what? Your nitrogenous bases. Your nitrogenous bases. So they are going to form the bond. But you have a very strong backbone, which is made up of the phosphate molecule. Because alternate amino groups in one polypeptide chain are hydrogen bonded with alternate carboxyl groups in the polypeptide backbone of the adjacent chain, in each polypeptide backbone, alternate carboxyl and amino groups are free to form hydrogen bonds. So the 3' the 5' backbones are free to form hydrogen bonds with their counterparts in different polypeptide backbones on the opposite side. So a B sheet or a beta sheet can consist of multiple aligned segments in the same or different polypeptide chains. Then for others, you should be informed that there are two main types of the polypeptide backbones, the 5' and the 3' phosphate. Other types of interactions are also important in protein folding, of the protein. For example, covalent bonds may form between the sulfur atoms of pairs of cysteines in different parts of the polypeptide. And cysteine is another type of what? Amino acid. We have varying type of amino acid. I'll really display some for you in the next few slides. So the rules of folding are so complex, except for the simplest proteins. For the simple ones, it is so easy to understand, but some it is very complex. The rules of engagement is very, very complex. So the final shape of a protein cannot usually be predicted from the amino acid sequence alone. Many protein molecules consist of more than one polypeptide chain. You have a varying, that is why it is referred to as what a polypeptide. You might have, that is why in some cases you may have a homozygous, and in some other cases you might have what, a heterozygous gene, whereby there is an interspersion. There is an integration of another gene. But when you have a homozygous, that means that it is 100% of one particular type of it. So when this is the case, the protein is said to contain subunits, and the subunits may be identical or different. For example, hemoglobin, the oxygen carrier of blood, consists of 4 subunits. 2 copies of each of the 2 different polypeptides are designated with the alpha chain and the B chain. So the use of alpha and beta and names of polypeptide chains has nothing to do with the alpha helices and B sheets that are found within the polypeptide. So here are a few examples of amino acids. You have arginine, and you realize that arginine, glutamic, lysine, aspartic acid, phenylalanine, valine, cysteine, leucine, methionine, isoleucine, tryptophan, choline, histidine, triosine, glycine. These are just a few. These are just a few. These are 15. In actual sense, there are 20 of them, and I cannot display all here. That is why I decided to take 3/4 the number of amino acids and display to you. Realize that they are different from each other. There's none which is same. There's none which is same here. They are different from each other. Phenylalanine, you realize you have a benzene ring in there. It's quite different from valine and cysteine. All of them are different. They are unique in their own molecular arrangement. So formation of
Doc.
Yes.
Please, I wanted to find out if they are all amino acid. Why the structure difference?
No. They are all amino acids, but they are different. They are different, right? Let me explain something to you. I don't know you, but I might not look like you. We might be of the same. We might be of the same height. Might be of the same weight, but we are even not the same. Even identical twins have what differences. That is how amino acids behave. But they are still embodied in living organisms. We have 20 different types of them. I couldn't display all. I was able to display 15 out of the 20. So they have specific roles each of them play in building up proteins and amino acids in living organisms, in prokaryotes as well as in eukaryotes. Is that clear?
Yes, sir. Doc.
Can I continue, please?
Yes, Doc.
Very well. So in here, there is formation of a dipeptide by reaction of carboxyl group of one amino on the left with another amino of a second on the right. Dipeptide simply means that a protein molecule made up of what? 2 bonds. There was a general amino acid that we looked at earlier. One with an imine group, the other with what? A carboxyl group. That means that we have 2 dipeptide. We have 2, okay, R groups in its formation. So you realize that you have 1 R and there is another R. And in between, you have that blue tincture there, okay? And it is going to merge. And where is it merging? It is merging at the epinos or amino terminus. That is where the merging occurs. And it is merging with what? The carboxyl group, the COOH. okay, the carboxyl group, that is where the peptide bond is going to be formed. That is where a peptide bond is going to be formed. So a molecule of water is eliminated to form a peptide bond. Why? Because we have what? A carboxyl group COOH and an amino group. The amino group also bears what? NH2. So you have an OH going to merge with what? One H from the amino to form what? Water. And that water is what? Eliminated. So that informs the dipeptide reaction of the carboxyl group of amino acid. So in here, we have the ribbon diagram of the part of a backbone of a polypeptide, showing the ways in which the polypeptide is folded. So you have a singleton, and gradually it folds up. So if you look at the arrows, it folds up, it is single, then it goes that way, it keeps on winding until, and one wind, you have what? Two phosphate groups, okay? One bearing what? Amino, and the other bearing a carboxyl. So they keep forming, twisting around each other until they form that ribbon or the helix in that order. So it keeps twisting until you have that twist. And as it is twisting around the amino as well as the carboxyl. And there is what? Elimination of water. Because when the bond forms, and the dipeptide bond forms, okay, water is eliminated from the bond, the peptide bond. So relations between genes and polypeptides. So that actually took a long time to find out that genes control structure of proteins. Archibald Garrow suggested that hereditary human diseases such as phenylketonuria result from inborn errors of metabolism. So this idea came in 1940s when George Beadle and Edward Tertune demonstrated using neurospectroscopy that genes govern the ability of the fungus to synthesize amino acids, purines, vitamins. So most genes contain the information for synthesis of only one polypeptide chain. Furthermore, the sequence of nucleotides in a gene determines the sequence of amino acids in polypeptide. So whatever amino acids is in an organism relates to the sort of genes that that organism actually possesses. So this point was first proved by studies by the tryptophan synthesis gene, TRPA, tryptophan synthase gene in Escherichia coli. That's a bacterium, and there's a prokaryote. So a gene in which many mutations had been obtained and accurately mapped. This was where they were able to relate genes with polypeptides. So the effect of the numerous mutations on the amino acid sequence of the enzyme were determined by directly analyzing the amino acid sequences, the wild type and the mutant type. So they investigated, let's say, the wild type of Escherichia coli, and they mutated it. They added certain genes to actually make it what? Robust or add otherwise. So each mutation was found to result in a single amino acid substituting for the wild type amino acid in the enzyme. And more importantly, the order of mutations in the genetic map was polypeptide chain, polypeptide chain in that regard. So this attributes of genes and polypeptide is called collinearity attribute of genes and polypeptide is referred to as collinearity. And it simply means that the sequence of base pairs in DNA determines the sequence of amino acid in the polypeptide in a collinear or point-to-point manner. The long and short of it is that the DNA molecule is made up of certain nitrogenous bases, and each nitrogenous base has a complementary base pairings. So they merge at certain points, and that is what collinearity is about, point to point. A adenine in that order will definitely merge or co-line with what? Thymine. Guanine in that order will definitely co-line with what? Cytosine. And that is the rule of the game. However, collinearity is found almost universally in every prokaryote, almost all prokaryotes. But on the other side, we will later see that in eukaryotes, non-informational DNA sequences interact the continuity of most genes. That is why, as a human race, we have a variety as a result of what? Diseases. That is why in some humans, some are unable to what? To speak. Some are unable to hear. Some are blind. And some have other diseases such as Down syndrome, a whole lot. So that informs the non-informational DNA sequences that interrupt continuity of most genes. And in these genes, the order of mutations along the gene correlates with the respective amino acid substitutions. So, in review, proteins are the molecules that catalyze most intracellular chemical reactions for regulating gene expression and for determining many features of the structures of cells, tissues, and viruses that are structural proteins. So protein normally is composed of one or more chains of amino acids. The 20 different amino acids commonly found in polypeptides can be joined in any number and in any order. I was able to display 15 to you, but in natural sense, there are 20. Each amino acid contains a carbon atom, the alpha carbon. Alpha simply means first. So to which is attached one carbonyl group, COOH. One amino group, that's the NH2, and a side chain commonly referred to as the R group. And the R group normally involves any other chemical group. Polypeptides are synthesized by adding successive amino acids to the carboxyl end of the growing chain. So the COH end or terminus is where other peptide bonds are added to it. And earlier I made mention of the fact that that is where water is what eliminated. Conventionally, the amino acids of a polypeptide chain are numbered, starting at the amino terminus. Most polypeptide chains are highly folded and a variety of three-dimensional shapes have been observed. So I made mention of the fact that just as you go about naming organic compounds, the longest chain, the naming of polypeptide chains begins with the amino group. You begin from the amino end, the amino term. Two fundamental polypeptide structures are the alpha helix and the beta sheet. So the alpha helix, represented as a coiled ribbon, is formed by interactions between neighboring amino acids that twists the backbone into a right-handed helix in which the amine each peptide group is hydrogen bonded with what? The carboxyl group in the peptide group located 4 amino acids further along the helix. So just as it is twisting, one twists the amine group and the other twists the carboxyl group through there. So in contrast, the B sheet or the beta sheet is formed by interactions between amino acids in distant parts of the polypeptide chains. And the backbones of the polypeptide chains are held flat and rigid, forming an alpha sheet. So because of the alternate amino groups in one polypeptide backbone that are hydrogen bonded with the alternate carboxyl groups in the polypeptide backbone of the adjacent chain. So they all twist in a swelling mode or fashion, and at the end of the day, you end up getting that ribbon-shaped or coiled polypeptide chain emerging. So that brings us to the end of the first session. Today, I have a few questions for you. So each and everybody, please pick up your jotters and answer these questions. I'll take them in the course of the week. If you are able to submit it by the close of the lesson, fine. But you submit it individually. The first question: what are proteins? Outline the relationship between proteins and amino acids. And you provide the chemical structures for the following amino acids: methionine, arginine, glutamic acid, aspartic acid, as well as lysine, valine, leucine, threonine, glycine, and histidine. So you provide the chemical structures of these amino acids. Okay. Then you briefly describe how polypeptides are formed. So please finish writing up this question before I continue to the next session. If I'm done, you prompt me, please. Yeah, answering everything.
Okay.
Sir, please, have we send it to your email or WhatsApp?
Yes, you can. You can send it to my. You have my email, right?
Sir,
Have my.
No,
You have
sir.
my email.
please, are we doing it now?
We don't have. No,
No,
sir.
sir.
You don't have.
So I will definitely let you. I'll definitely let you have it at the close of the lesson. Okay. So what about my number? WhatsApp. Don't forget to
Wait, your number. We have your number.
attach your registration numbers. Is that right? Because it is a mode of assessment. That is what I'm going to use to assess you.
Okay, sir.
Okay, so colleague, just like
Very
we
well.
did for biotech, the same way we are doing this one. You add your details, then you forward it to the mail that he will provide. So let's move on. So I think we can move on. We have screenshot their questions. We'll do it later on and then send it to you.
Very well. Thank you very much.
In course of the
All right. So section 2 has to do with transcription. And with this, you are transcribing something. It's just like you are transcribing an interview. Okay, transcribing an interview, maybe during your analysis, doing your project work, you pick up some data and you need to transcribe. So the genes and proteins also undergo this form of analysis. You need to transcribe it. Transcription. So by the end of this session, you should be able to explain the transcription process and discuss the chemistry as well as the characteristics, structure, and classes of prokaryotic RNA. So let's look at the general features of an RNA synthesis. Earlier, before I delve into it, I always make mention of the fact that it is out of the DNA that an RNA emerges. So you will definitely have complementary base pairings of the various nitrogenous bases on the DNA before an RNA emerges. So the DNA would be sitting on the powerhouse of the cell in the nucleus. Then it will undergo the DNA polymerase would act on it. It will unwind and release the complementary base pairing into the cytoplasm. And that is carrying some information. And that informs the messenger ribonucleic acid, mRNA. So the mRNA is just a direct copy of the mother DNA, which is undergoing some process in the nucleus. And so it releases it into the cytoplasm. And from there, there are other forms of translations going to take place. So before it leaves the nucleus, you realize that thiamine, which used to sit on the DNA nitrogenous base, will be replaced with uracil. That informs it. So you would have adenine, guanine, cytosine, and thymine will be replaced by what? Uracil. And there is no way you would find thymine on the RNA molecule. So the first step in gene expression is the synthesis of an RNA molecule copied from the segment of DNA that constitutes the gene. And the essential chemical characteristics of enzymatic synthesis of RNA resemble those of the DNA synthesis as described, as I've thoroughly explained to you. So the process is as summarized. The precursors in the synthesis of RNA are the 4 ribonucleotide and 5 triophosphates. So namely, they are the adenosine triphosphate, A triphosphate, adenosine triphosphate, ATP, guanosine triphosphate, GTP, cysteine triphosphate, that's CTP, uridine triphosphate, UTP. So you realize that there's no thymine triphosphate. Thymine is replaced here. That is why we have what? Uridine triphosphate. So they differ from DNA precursors. RNA differs from DNA precursors. The long and short is that RNA normally occurs. There's no way you find an RNA in the nucleus. You'd always locate it in the cytoplasm. So they differ from the DNA precursors only in that the sugar is ribose rather than what? Deoxyribose. DNA precursors, RNA precursors, they differ from the DNA precursors only in that their sugar is ribose sugar. That is why it is referred to as what? Ribonucleic acid. And DNA is referred to as what? Deoxyribonucleic acid. And the base uracil replaces thymine as discussed. So you have deoxyribose, that's DNA, okay? That's old in ribose, okay? The hydroxy molecule in ribose of what? An RNA, okay? Then the hydrogen in uracil, you have thymine. Differences in structures of ribose and deoxyribose, and in those of what? Uracil and thymine. So you have thymine occurring here. That's a phenyl ring, okay? Phenyl ring, benzene ring, thymine. And you have an OH in the deoxyribose. You have OH, that is water. Then there is another, and you realize that it is labeled. Okay, it is labeled. You have numbers 1, 2, 3, 4, 5. With regards to the carboxyl group, okay, that is why it's been. Hello. Please, you are giving us a feedback. Kindly mute for us. So that informs the differences in structures of ribose and deoxyribose nucleotides. Okay, right? Where one is what? A deoxyribose, and the other is a ribose sugar. These are sugars. Okay. Distinction between what? DNA and RNA. Distinction between DNA and RNA. So the general features of RNA synthesis, RNA formation. What are the general features? So in the synthesis of RNA, a sugar phosphate bond, that is informing its backbone, is formed between the 3' hydroxyl group, a 3' hydroxyl group of one nucleotide and the 5' triphosphate of the next nucleotide in line. So this is the same chemical bond as in the synthesis of DNA. But the general features, the same thing that occurs with the formation of DNA definitely occurs in here too. But just that in RNA, you have a ribose sugar. And in DNA, you have a deoxyribose sugar. So this is the same chemical bond as in the synthesis of DNA, but the enzyme is different. The enzyme used in transcription is RNA polymerase rather than DNA polymerase. So the only difference here is the enzyme, which brings about the formation of what? RNA. You have RNA polymerase instead of what? DNA polymerase. So the linear order of bases for this one, I keep saying the linear order of bases. A will definitely pair with U in RNA. In DNA, it would have been A to T, and in RNA is going to be. So the linear order of bases in an RNA molecule determined by the sequence of bases in the DNA template. So each base added to the growing end of the RNA chain is chosen for its ability to base pair. So the bases will be C, T, G, and A in DNA strand, and cause G, A, C, and U respectively to be added at the end of an RNA molecule. So in DNA, you have what? E to T, G to C. G to C that changes in both nucleic acids, but the T is replaced with a U in RNA. Thymine is replaced with uracil in RNA. Nucleotides are added only to the 3-hydroxyl end of the growing chain as a result of the 5' end of the growing RNA molecule bearing a triphosphate group. So you know that the 5' to 3' direction of RNA chain group is the same as that in what? DNA synthesis. So let's look at the general features. And a significant difference between DNA polymerase and RNA is that RNA polymerase is able to initiate chain group without a primer. But chain group occurring in DNA have to commence with a primer. So furthermore, each RNA molecule produced in transcription derives from a single strand of DNA. In the preamble before I started session 2, I made emphasis to the fact that the DNA is sitting in the nucleus and there has to be a mirror copy. You have to unwind and release one strand. That double helix structure has to release one strand, forming the messenger RNA into the cytoplasm, where it has to bind with other ribosomes to undergo protein synthesis and that order. Each RNA molecule produced in transcription derives from a single strand of DNA, because in any particular region of the DNA, only one strand serves as a template. So one strand would remain in the nucleus and begin forming that template to become a DNA molecule again, double helix structure again. It will undergo a period of what? Metaphase. Then the meiotic or meiotic process would commence again. Meiosis 1, meiosis 2 would what? Commence again. So the synthesis of RNA can be described as consisting of 4 discrete stages: a promoter recognition stage, a chain initiation stage, chain elongation. Chain. Every chain is made up of what? Certain segments. That the chain is short or long. Okay? And for how long can it be? You can't have a chain that goes around the whole world. It keeps coiling. It definitely has to end. And that informs the chain termination stage. We'll be taking it one after the other and look at what goes into promoter recognition, chain initiation, and chain elongation, as well as chain termination. Chain termination means that it has come to an end and there is a terminus, and that ends the nucleic acid or polypeptide chain. So the promoter structure of a prokaryote, showing the consensus spaces and the tata box. So in here, like I said earlier, a mirror copy of the DNA informs the messenger ribonucleic acid, mRNA, and that is the promoter. Is going to promote the formation of other ribonucleic acids. And the other ribonucleic acid, you have messenger RNA, and that is the promoter. Then you have the transfer RNA, as well as what? The ribosomal RNA or aminosylated RNA. So you realize some figures, -30, -10, the transcription starts site, that's +1. And you see a 5', okay? A 5' on the mRNA. And you have some nitrogenous bases on the mRNA. You have adenine, you have uracil, as well as what? Guanine. As well as guanine. So it's complementary base pairing. You definitely have to get what? A uracil blending with the adenine, then an adenine blending or merging up with the uracil. And guanine would always pair with one cytosine in that jurisdiction. So you have a promoter structure that's -30 region. You have T, T, G, A, C, A, C, T, G, and this is in the DNA, okay? Because this is what occurs before the structure actually what unwinds, before DNA polymerase strikes it, divides it into two, and one leaves into the cytoplasm. So this is a promoter structure of a prokaryote, showing the consensus sequences in the Tata box. The Tata box is the one in between, okay? The first and the one below. Pre-brown box. Okay, so you have T, you have 82, 84, G. The consequences, they are in percentages. Then we have inverted repeats. Inverted repeats, DNA. So you have that double helix structure. And the direction of transcription goes in that order. That means that one is going to the 3' end is the one which is going to move into the cytoplasm, whilst the one on top, the 5' and the inverted repeats will stay in the nucleus, and other complementary base pairings would again merge and it forms another giant structure. So direction, transcription, okay, the template strand, you have the various complementary base pairings as labeled in there. Then the terminus of messenger RNA, okay, it begins as DNA polymerase act on it. It begins to what? Unwind. It begins to unwind, and after it's unwinded, it moves into the cytoplasm. So the base sequence in A, it informs the base sequence of the transcription termination region for the set of tryptophan synthesizing genes in what? Escherichia coli, which is the prokaryote bacterium cell. So the inverted repeat sequences, which is blue, okay? The inverted repeat that is in blue are characteristic of termination sites. These are termination sites. They leave. They are possible. Possible sites of what? Leaving. These are the possible sites of leaving. They leave and move into the cytoplasm. And B, the 3 terminus of RNA transcript that is folded to form a stem and loop structure. Okay, a stem and loop structure. So as it has unwound, this informs the messenger RNA. Messenger RNA. This is a direct copy of the DNA molecule. Okay. So messenger RNA, amino acids do not bind directly to DNA, but they need to bind to the messenger RNA in order to form the DNA. So consequently, intermediate steps are needed for arranging the amino acids in a peptide chain in the order determined by the DNA base sequence. So this process begins with transcription of the base sequence of the template strand of DNA into the base sequence of an RNA molecule. In prokaryotes, this RNA molecule is referred to as the messenger RNA or mRNA. it is used directly in polypeptide synthesis. So in eukaryotes, the RNA molecules generally process before it becomes mRNA. So you should know the distinction between the process, the synthesis occurring in eukaryotes, as well as the synthesis occurring in what eukaryotes. This point is a very crucial point that you have to take note of. In prokaryotes, this RNA molecule is called what? Messenger RNA, or mRNA, and it's used directly in the polypeptide chain synthesis. But in eukaryotes, the RNA molecule is generally processed before it becomes what? RNA.
Wow.
Hello? Hello? Please kindly mute, because you are giving me a feedback here, please. Are you asking a question? All right. So the amino acid sequence is then determined by the base sequence in mRNA by the protein synthesizing machinery of the cell. And not all base sequences in an mRNA molecule are translated into the amino acid sequences of polypeptides. Example, translation of an mRNA molecule rarely starts exactly at one of its ends and proceeds to the other end. Instead, initiation of polypeptide synthesis may begin many nucleotides downstream from the end of the mRNA. So as we looked at it in the previous diagrams, how the promoter begins from the DNA, it's informing the messenger RNA, and the messenger RNA sends a direct information, or sends the complementary base pairings of the DNA into the cytoplasm for protein synthesis to occur. So the untranslated 5' segment of RNA is called a leader, and in some cases contains regulatory sequences that affect the rate of protein synthesis, because it is carrying information directly from the DNA. So the leader is followed by a coding sequence, also called an open reading frame, OR, which specifies the order in which the amino acids are present in the polypeptide chain. A typical coding sequence in an mRNA molecule is between 500 and 3,000 bases long. So you can imagine how long this base pairings are. 500 and 3,000 bases long, depending on the number of amino acids in the protein. So like the leader sequence, the 3' end of an mRNA molecule following the coding sequence is not well translated. The template strand of each gene is only one of the 2 DNA strands in the gene. So the DNA is a double helix structure. So when it unwinds, when DNA polymerase acts on it, that is when it unwinds. The hydrogen bonds are broken. One leaves the terminus. One leaves, and as it is leaving, it is carrying a mirror copy and mirror image of the parent DNA into the cytoplasm. But which DNA strand is the template can differ from gene to gene along the DNA molecule. So that is except in some small viruses. And not all mRNA molecules are transcribed from the same DNA strand. These are exceptions. So in an extended segment of a DNA molecule, mRNA molecules will be seen going in either of 2 directions, depending on which DNA strand functions acts as a template. These are exceptions, but the norm is that it carries the exact information on the DNA into the cytoplasm. In prokaryotes, on the other hand, just as I said earlier, with the crucial point, they have to undergo what? They just don't need that processing in eukaryotes as in the prokaryotes, because they are made up of what? A nuclear material. They don't have a true nucleus. So in prokaryotes, most mRNA molecules are degraded within a few minutes after synthesis, because it's just one cell. It's just one cell. So in eukaryotes, a typical lifetime is several hours, although some last only minutes and others persist for days. So that is what occurs with messenger RNA synthesis in prokaryotes as well as in eukaryotes. So in both kinds of organisms, the degradation enables cells to dispose of molecules that are no longer needed. So some molecules are not so vital, just have to discard it and carry on with the ones which are vital. So the short lifetime of prokaryotic mRNA is an important factor in regulating gene activity. So this diagram here, you have RNA transcript, you have the promoter site, then the complementary DNA strands, origin and direction of transcription, you have transcriptional termination site, a typical arrangement of promoters and termination in a segment of DNA molecule. So the promoters are present in both DNA strands. Promoters, they are present in both DNA strands. So this is a DNA, one 5' terminus and the other 3'. Okay, so the 3' is the one on top, and the 5', because it's the 3' which leaves the nucleus site, informing the mRNA. So termination sites usually located such that transcribed regions do not overlap. Transcribed regions do not overlap. So in RNA, polymerase of bacteria is a multi-subunit protein, prokaryotic RNA polymerase structure. All of the classes of RNA in bacteria are transcribed by a single RNA polymerase. So the enzyme that acts on the segregation of RNA is the RNA polymerase. An enzyme together with the cofactors required for activity is called a holoenzyme or haloenzyme. An apoenzyme is an inactive enzyme. Activation of the enzymes occurs upon binding of an organic or inorganic cofactor. A holoenzyme is an apoenzyme together with its cofactor. The multi-subunits of the RNA polymerase and their roles are summarized below. So you have subunits, row number 1, you have alpha, and alpha has 2, beta 1, beta prime 1, gamma 1. These are on certain forms of phosphodiester bonds binding DNA templates, which recognizes promoter and facilitates initiation. So alpha 2, beta prime gamma goes into alpha 2 beta beta prime plus the gamma, the sigma factor core polymerase, what actually informs a holoenzyme. You have a core polymerase and a sigma factor coming together to form the holoenzyme. So the multi-subunit RNA polymerase showing its core polymerase and the sigma factor. Functions of the sigma factor. So the sigma factor in RNA polymerase has the following functions. it serves as the subunit of RNA polymerase, and this is an initiation factor. Several different sigma factors in Escherichia coli that are specific for different sets of genes. Again, sigma factor functions to ensure that RNA polymerase binds stably. It binds in a stable mode to DNA only at the promoters. So it is because of the sigma factor, that is why when the DNA polymerase releases the messenger RNA, it's as a result of the sigma factor that parent strand, the template which remains behind, is able to form another lead structure and back to a DNA again. So sigma destabilizes non-specific binding to non-promoter DNA. So those that are not supposed to bind, like the uracil, that is not supposed to bind with an adenine in the nucleus, would never occur. It's as a result of the sigma factor to relegate it to the background and bring in a thymine to bind with the adenine. Sigma stabilizes specific binding to promoter DNA. Then it also accelerates the search for promoter DNA. So these are the core functions of the sigma factor. Now let's look at the sigma cycle. It first helps RNA polymerase to find a promoter by binding to the -10 region. And this is only moderately stable as a closed duplex. DNA is double-stranded duplex. This is only moderately stable as a closed complex, but becomes highly stable as an open complex. Highly stable after it has unwound. Okay, it becomes highly stable because it's standing alone in the nucleus. Sigma factor binding helps to denature the adenine thymine rich that's cribnal box forming the open complex. It helps to denature it. It helps to destroy the hydrogen bond. So once initiation takes place, sigma factor dissociates, freeing the core RNA polymerase from the very high affinity interaction at the promoter and allowing it to move away from the promoter to transcribe the rest of the genes. So it's as a result of the sigma factor that allows RNA to form. So without the sigma factor and cycle, RNA cannot be formed. The sigma factor is then free to rebind other core RNA polymerase enzymes. So that is what informs the messenger, the transfer, as well as the ribosomal RNA or aminosylated RNA. So let's look at what occurs. Closed promoter complex, this close, so moderately stable. So the sigma subunit binds to the -10 region. RNA polymerase holoenzyme, that's positive gamma factor, it opens the promoter complex, which is highly stable. Then the holoenzyme has a very high affinity for promoter regions because of the sigma factor. Because it is RNA formation that is about to occur. If DNA doesn't unwind, RNA wouldn't form. So when it unwinds, then it comes in the holoenzyme. So once initiation takes place, RNA polymerase does not need very high affinity for the promoter. Once it has unwind, there's no need to accumulate so much energy to bring about the process again. Okay, so meiosis 1 before the resting stage, the interphase, that is where all of these occur. And after it has finished occurring, prophase, those other stages that inform meiosis 1 and 2 roll in that order. When prophase 1, it actually entails the long processes, the lectitine, the diplotin, those 5 stages that occur before it moves on to the metaphase stage. So sigma factor dissociates from the core polymerase after a few elongation reactions. So this is sigma, it can rebind other coenzymes. So the sigma cycle and the role it plays in RNA polymerase binding. So at an instance, you see that it is all straight. Then it begins to bulge. It begins to bulge. Then there is a minus, okay, that releases the gamma, the sigma. Then sigma can what? Rebind. Can rebind. That informs the elongation state. So the sigma cycle and the role it plays in RNA polymerase binding. So the first step in gene expression is the synthesis of an RNA molecule copied from the segment of DNA that constitutes the gene. The essential chemical characteristics of the enzymatic synthesis of RNA resembles those of DNA synthesis. A significant difference between DNA polymerase and RNA polymerase is that RNA is able to initiate chain growth without a primer. And again, each RNA molecule produced in transcription derives from a single strand of DNA, because in any particular region of the DNA, only one strand serves as a template for RNA synthesis. So the synthesis of RNA can be described as consisting of 4 discrete stages, namely the promoter region, the chain initiation, chain elongation, and chain termination. Again, RNA polymerase binds to DNA within a base sequence from 20 to 200 bases in length, called promoter. So the more closely the promoter elements resemble the consensus sequence, the stronger the promoter. In eukaryotes, the promoter sequences are also other DNA sequences called cances that interact with the promoter to determine the level of transcription. In prokaryotes, most mRNA molecules are degraded within a few minutes after synthesis. In eukaryotes, ethical lifetime is several hours, although some last only minutes, and others persist for days. So in both kinds of organisms, the degradation enables cells to dispose of molecules that are no longer needed. So the short lifetime of prokaryotic mRNA is an important factor in regulating gene activity. So that brings us to the end of Unit 4, Session 2. We are now moving on to Unit 4, Session 3, and we'll be looking at RNA processing. RNA processing. How is RNA processed? The various stages in which RNA is processed is what we'll be looking at. So by the close of this session, you should be able to explain the major steps in eukaryotic mRNA processing. Then you should also be able to understand and explain the concept of splicing. Splicing. What is splicing? What at all is splicing? So we'll be looking at all the major steps involved in eukaryotic mRNA processing. And understand and explain the concept of splicing. All right, so let's look at capping, cleavage, and polyadenylation. Capping, cleavage, and polyadenylation. So each end of the eukaryotic transcript is processed, each end, and we are informed of the ends that eukaryote and has. So the 5' end is altered by the addition of a modified guanosine 7-methyl guanosine in uncommon 5-5 instead of 3-5 linkage. Okay, so in capping, right? Instead of what 3-5, you have what a 5-5, and that informs what a 7-methyl guanosine end with regards to its linkage. And this terminal group is called a cap, because it is made up of what the same end, 5 prime, 5 prime, instead of the normal 3-5. So because of the infusion of what a modified guanosine, referred to as 7-methyl guanosine, being infused into the end, the nitrogenous, the phosphate end, and the backbone. The 3' terminus of a eukaryotic messenger RNA molecule is usually modified by the addition of polyadenosine sequence and the poly-A tail out of many as 200 nucleotides. So cleavage of the primary transcript occurs approximately 10 to 30 nucleotides at the prime word of AAU consensus site. And so in the other terminal end, with regard to the 3' terminus end, there is also an addition of what a polyadenosine sequence. Unlike in the first one that we talked about, guanosine, modified guanosine. This one, it is a polyadenosine. So you realize that you have more of the A's being substituted, and there are about 10 to 30 nucleotides of them. 10 to 30 of them. So polyadenylation catalyzed by polyadenylation polymerase. So approximately 200 adenylate residues are added. The 5' is necessary for the mRNA to bind with the ribosome to begin protein synthesis. And the poly-A tail helps to determine the messenger RNA stability. RNA splicing. RNA splicing. A second important feature peculiar to the primary transcript in eukaryotes is the presence of segments of RNA called introns. Introns. Okay, so these are introns or intervening sequences that are excised from the primary transcript. Introns are already embedded. That is with regards to eukaryotes. So accompanying the excision of introns is a rejoining of the coding segments, that's exons. Recoding. The ones that are able to join it from outside, like the ribosomes, the transfer RNA pick up certain ribosomes from the cytoplasm, and they are referred to as the exons to form the mRNA molecule. So the excision of the introns and the joining of the exons is called RNA splicing. Excision of the introns and the joining of the exons is called RNA splicing. So the mechanism of RNA splicing is illustrated schematically in the figure below. So let's look at it. Upstream, you have a donor splice site, and you have a branch site, acceptor site, splice site, and another downstream. So we have upstream, downstream, upstream, downstream. Then we have spliced exons below. Okay, we have spliced exons below. So the A of branch sites, A of branch sites, that's the adenine of branch sites, attacks the guanine at the donor splice site. So there is a movement from the branch site. Okay, adenine from here attacks the cytosine at the donor splice site. Then it attacks the guanine, sorry, is G. A attacking the G at the donor site. A attacking the G at the donor site. So A to G linkage is 2' to 5'. This is not the normal 3 to 5' because it is being spliced. Introns are being exercised with the exons. Okay, so you realize that the splice site, the exons, react. You have 5' to 3'. Then it rapidly digests to small pieces. Okay, a schematic diagram showing removal of 1 intron from a primary transcript. The A adenine nucleotide at the branch site attacks the terminus of the 5' exon, cleaving the exon intron junction and forming a loop connected back to the branch site. So you have a street that is downstream, 5' towards the downstream, okay, and one portion referred to as the branch site, okay, it's attacking the donor splice site, and that informs the 5'2'. 5'2'. It's not normal. A attacking G linkage is what, 2' to 5', and not the normal phosphate 3', 5' phosphate backbone that occurs. This is not actually normal. So the spliced exons or splice sites form a loop. So this is the loop that is forming. Okay, it's that it is coiling, right? You have some of the nitrogenous bases, and it forms a loop to the small pieces, rapidly digested to small pieces. Then it moves on to the 3', So in the first step of splicing, the 2' hydroxyl, okay, OH of the adenosine at the branch site, which is located a short distance upstream from a run of the pyrimidine Y. And you should know that we are using 2 nitrogenous bases, 2 main nitrogenous bases, the purines and the pyrimidines. Near the acceptor site attacks the phosphodiester bond at the donor splice site junction. And the attack results in cleavage. Attack results in cleavage at the donor site and formation of a branched molecule known as the lariat, because it has a loop and a tail. It has a loop, it coils, and at the same time has a tail. So the AG linkage at the knot of the lariat is unusual in what, 2' to 5'. It doesn't occur, but it has occurred instead of the 3 to 5'. So in the final step of splicing, the 3-OH of the guanosine of the donor exon attacks the phosphodiester bond at the acceptor splice site, freeing the lariat intron and joining the donor. So the acceptor exons together. So the lariat intron is rapidly degraded into individual nucleotides by the nucleases. So RNA polynuclease informs this formation. So you realize that the 2' would occur. That isn't normal. But after the 3-hydroxy of the guanosine attack the phosphodiester bond, the acceptor splice site, it frees the lariat. The lariat is the 2 to 5 prime intron and joining the donor and acceptor exons together. So that informs the 3:5 direction that inform the nucleotide by the nucleases. So RNA splicing takes place in nuclear particles known as what? Spliceosomes. So it always occurs in what? The nucleus. There's no way this thing is going to occur in the cytoplasm. DNA's house or its place of abode is in the nucleus. There's no way you would find what? DNA in the cytoplasm. So these abundant particles are composed of proteins and several types of specialized small RNA molecules ranging from 100 to 200 bases. And the RNA molecules that you would always find in the nucleus are the messenger RNA because they are carrying the exact mirror copies, the parent copies of the DNA into the cytoplasm. That is why they are referred to as messengers. Messengers. So the specificity of splicing comes from the small RNAs, some of which contain sequences that are complementary to the splice junctions. But numerous spliceosome proteins are also required for splicing. One model for the process is illustrated in the next diagram, in which the Russell 1, Russell 2, and 5 are designations for 3 different types of small nuclear RNAs. So the ends of the intron are brought together by the U1, that Russell 1, which forms base pairs with nucleotides in the intron and go to the 5' and 3' ends. So the ends of the exons are brought together by the Russell 5, which forms bases or base pairs with nucleotides in the exons at both the donor splice site and the acceptor splice site. So the intermediate produced by the first trans-esterification reaction involves a branching polynucleotide where the adenosine makes bonds with both its true OH and its OH. So you realize that after it has unwound or unzipped the DNA, there are lots of complementary base pairings on the other strand. And with regards to the DNA, because of the DNA polymerase or the hydrogen bonds that form, the bonds are very strong and they bond each complementary base together. So with regards to the messenger RNA, it is not all the complementary base pairings that will be bonded on the strand. Okay, it's just a few that might pick up certain base pairings and continue. It breaks. So that informs the 2-hydroxyl, the 2' hydroxyl end and the 3' hydroxyl end. So the second cleavage ligation reaction occurs by attacking the 3' hydroxyl at the 3' end of the upstream exon on the 3 end of the intron. So this simultaneously releases the intron transcript, and it's called the lariat, and ligates the 2 exons together. I know that the branch site is held in place by what? The U2. That's O2. So here we go. The model for RNA splicing: U, 1, 2, and 5 are small nuclear RNA molecules. So we have U1, we have U2, and U5. So as it has unwound, this is just one. Okay, this is just one. We have what? A 3' end and a 5' end. The 3' end is the what? Intron. That is what is going is going, leader. Okay, and the exon is what exists. The terminus, the terminal end. Okay, so you'd realize that you have the various complementary base pairings in there. You have U, that is for uracil, because it has been replaced. Thymine has been replaced by uracil. You have G or C. Okay, you have U, C, A. You have A, you have U, you have G, in that order, because we are referring to what? Ribose sugar. So thymine has been replaced, and uracil is what is checked here. So A at branch site attacks G at donor splice site, as indicated earlier in the figure above. So the overall processes involved in transcription are similar in prokaryotes and eukaryotes, but there are major differences in relation between the transcript and the messenger RNA used for polypeptide synthesis. So in prokaryotes, the immediate product of transcription is mRNA, whilst the primary transcript in eukaryotes must be converted into mRNA. In prokaryotes, no processing occurs in the RNA. It can even start to be translated before transcription is complete. In eukaryotes, RNA is synthesized by RNA pool 2 as longer precursors, that's pre-mRNA of what mRNA, as a precursor of the mRNA. So processing of eukaryotic RNA involves 4 events: capping, cleavage, and polyadenylation, splicing, and methylation. In eukaryotes, there is the presence of segments of RNA called introns or intervening sequences. The excision of the introns and the joining of the exons is called RNA splicing. So that brings us to the end of Unit 4, Session 3. And you pick up this question again. 1. Explain why RNA processing is important. This assignment 2. Then explain the processes involved in eukaryotic RNA processing. 3. What is capping? Then finally, why does transcription start without processing? Let me add another question. 5, right? Question 5. What is cleavage? So you have 5 questions. The second assignment. So you submit it individually with your details, just as your course has duly admonished you. So when you are done, you prompt me for me to continue, please.
Doc.
Hello. Can I continue, please?
Please,
Yes,
I want to ask whether
yes,
that
please. We're taking a screenshot of it so you can continue.
Two people are talking at the same time. One at a time, please.
Doc, I was saying that we've taken a shot of it, but should this one be a continuation of the first one, or it should be
No, this is
separate?
a different assignment. Okay. So I've given you two assignments, which is going to be scored differently, and so that use it as a mode of assessment for you, please. The first one is assignment 1, and this is assignment 2. So you separate them, put it together, please.
All right, Doc.
Very well. On that note, can I continue, or I should wait on you?
So we're taking screenshots. We can continue.
Very well, please. So, Unit 4, Session 4 is on translation. Unit 4, Session 4 is on translation. By the end of this session, you should be able to explain the major steps involved in translation, understand and explain the factors required for translation to occur. So, let us start. Messenger RNA. It is needed to bring the ribosomal subunits together and to provide a coding sequence of bases that determines amino acid in the resulting polypeptide chain. So, just as I kept saying in the previous sessions, the DNA structure is a double helix, and before it undergoes the division, the hydrogen bonds needs to be broken so that one acts as a template, one strand needs to act as a template, whilst the other leaves as a messenger to join other subunits or cell inclusions in the cytoplasm for protein synthesis towards commence. And there are lots of ribosomes in the cytoplasm. There are no way you find ribosomes in the nucleus. So this is a nuclear reaction which takes place by releasing one strand of the genetic DNA molecule into the cytoplasm. So messenger RNA carries direct information of the DNA into the cytoplasm. So it is there that various cell inclusions, such as the ribosomes, would also find or merge up with certain portions of the nitrogenous bases to begin the process of what protein synthesis and formation of other proteins or amino acids. So that informs the messenger RNA, the work of the messenger RNA. Now let's look at ribosomes. These components are cell inclusions. They are components or particles on which protein synthesis takes place. Ribosomes. They are like some free agents. They are moving. Like you looking for an accommodation in Accra. It's so difficult, but you need agents. They have taken it upon themselves to locate homes that are going on rent, and they have their fees. So the same thing occurs here. The ribosomes are located in the cytoplasms, and they are waiting on what? Certain nitrogenous bases from the messenger RNA to undergo what? Protein synthesis, to form certain amino acids. So they move along an mRNA molecule and align successive transfer RNA molecules. Okay. On the messenger RNA molecule, they will align, the ribosomes will align on transfer RNA molecules. So the amino acids are attached one by one to the growing polypeptide chain by means of peptide bonds. So as they align themselves, they form what? Peptide bonds, protein bonds. And as we discussed earlier, the bonds are huge. They are running into hundreds to thousands of the various 20 amino acids that are supposed to be bonded. So the amino acids are attached one by one to the growing polypeptide chain by means of peptide bonds. So ribosome consists of two subunit particles, and the main subunit particle are the carboxyl and the amino group. Okay. So in E. coli, for instance, E. coli is a bacterium cell, and again a prokaryote. They don't have a true nucleus, but they have what the nuclear material. That is why most of the RNA that is formed are temporary. Okay. In E. coli, their sizes are 30S, and the 30S is referred to as Vedic. The unit is Vedic. He actually researched into that. And the sizes are 30S and 50S, the large subunit. So the counterparts in eukaryotes are 40s and 60s. In prokaryotes, it's what? 30 and 50. But in eukaryotes, those with true nucleus, organisms with true nucleus, that is both plants and animals, they are 40s and 60s. The S stands for Vedic unit. Vedic unit, which measures the rate of sedimentation of a particle in a centrifuge. So it's an indicator of size. So together, the small and large particles form a functional ribosome. The small and large particles, they form a functional ribosome. That is when they have aligned themselves to their complementary mRNA or tRNA. Okay. We have transfer RNA as well as what? Messenger RNA. Then when the ribosomes align themselves, they form ribosomal RNA. They form ribosomal RNA. Transfer RNA or tRNA. So the sequence of amino acids in a polypeptide is determined by the base sequence in the mRNA by means of a set of adapter molecules. So they adapt. Some of the molecules adapt, and they are referred to as the tRNA molecules, each of which is attached to a particular amino acid. So the messenger carries wholesale information from DNA, and whilst carrying it, they relay it through the transfer RNA, and the transfer also relays it to the ribosome. So each group of these three adjacent bases in the mRNA forms a codon. Three nitrogenous bases. They form codes. Code AUG, UGG, CCC, GGG, AAA, UUU. In that order, they form codons that bind to a particular group of three adjacent bases in the tRNA, and that is referred to as an anticodon. So they are bearing codes, and those codes are referred to as codons. Then they will align themselves to the adjacent bases in the tRNA, and that is referred to as anticodon, bringing the attached amino acid into line for addition to growing a polypeptide chain. Amino acyl transfer RNA synthesis. Amino acyl transfer RNA synthesis. So this set of enzymes catalyzes the attachment of each amino acid to its corresponding tRNA. This set of enzymes, the tRNA synthesis, okay, it catalyzes the attachment of each amino acid to its corresponding tRNA molecule. So a tRNA attached to its amino acid is called an aminosylated tRNA or charged tRNA. Aminosylated tRNA or a charged tRNA. So that is where in some books you'd come across ribosomal RNA because they bear some proteinous ends, amino ends, aminosylated tRNA or charged tRNA. So initiation, elongation, and termination factors, just as we discussed earlier with the translation. So these are the key factors that play roles in the whole translation process. For you to do something, you need to plan. And how are you going to process it? Then finally, you need to end. In any project or in any proposal, you need an introduction, then the methodology, then maybe your timelines, how you're going to go about it, bring it to a successful end. So let's take protein synthesis. Polypeptide synthesis can be divided into what? 3 stages. Protein synthesis is the same as that. And that involves the initiation, the elongation, as well as termination. Polypeptide synthesis is divided into 3 stages: initiation, elongation, as well as termination. Each stage requires specialized proteins. Initiation. The energy that you need to begin something is not the same energy that you need to finish. Then in prokaryotes, all of the components for translation are present throughout the cell. In eukaryotes, they are located in the cytoplasm, as well as in mitochondria and chloroplasts. Protein synthesis. A codon is a sequence of 3 DNA or RNA nucleotides, just as I said earlier. The nitrogenous bases, purines, pyrimidines, are bearing certain codes: GUC, AUC, CCC, GGG. It corresponds with specific amino acid or stop signals during what? Protein synthesis. An anticodon, on the other hand, is a sequence of 3 nucleotides forming a unit of genetic code in a transfer RNA molecule. So anticodons is born on the transfer RNA molecule, corresponding to a complementary codon in messenger RNA. So messenger RNA or mRNAs carry codons, and it bears on what the transfer RNAs would carry anticodons to bring about what the synthesis of protein, formation of proteins or amino acids. Initiation of protein synthesis. So in the initiation of translation, 2 initiation factors, IF1 and IF3. Initiation 1, initiation factor 1, initiation factor 3. They interact with 30s. And when they are interacting with 30s, 30S are Sveberg 30 subunits. you should know that it is occurring in what? Prokaryotes. It is occurring in prokaryotes. Organisms not bearing what? True nuclei. But if it is in the 50s and 60s, 40s and 60s, that means that they are occurring in what? The eukaryotes. So initiation factors 1 and 3 interact with 30 subunits at the same time. That another initiation factor 2 binds with a special initiator, tRNA charged with methionine. So in prokaryotes, the initiator, that is tRNA, actually carries formyl methionine, including the tRNA. And these components come together and combine. Somebody is giving me a feedback, please. Yes, thank you. So this is what occurs in the initiation stage of what? Protein synthesis, whereby initiation factor 1 and 3 interact with 30 Sferberg subunits at the same time. And that another initiation factor 2 had to bind with a special initiator charged with methionine, formyl methionine, to yield what? The transfer RNA. So these components come together and combine with an mRNA. The mRNA, earlier on, I made mention of the fact that it is carrying what? The codons, whilst the tRNA is bearing what? The anticodons. So in prokaryotes, the mRNA binding is facilitated by hydrogen bonding between the 16S Sferberg unit. RNA present in the T subunits and the ribosome binding site of the mRNA. In eukaryotes, the 5 cap on the mRNA is very instrumental. So together, the 30, the tRNA and formyl methionine and mRNA complex recruits 50S subunits in which the tRNA formyl methionine is positioned in the P site, then aligned with AUG initiation code, forming the 70 initiation complex, forming the 70 initiation complex. So let's look at what occurs in the initiation complex. You have messenger RNA and you have some ribosomes. Okay, so initiation factors, you have anticodon, then the codon. The codon is being born on the messenger RNA, mRNA, and you have anticodon. The anticodon is being born by what? The transfer RNA. Okay, so the transfer RNA bears an anticodon. Then in the all these is occurring in the cytoplasm. Okay, in the cytoplasm where you have these cell inclusions, right? The ribosomes. And so ribosome will be swimming freely to align itself to the codon to form the amino acid that it so desires, or to form the protein that it so desires. So you have a ribosome, that's a small subunit, that's 30S, that as indicated. Then you have the ribosome large unit, that's the 50S. Okay, you have an amino cell site. Amino cell site simply bears an NH2 site, peptidyl site bearing proteinous site. Then you have an edit site, EPA. And the ribosome would what? Attach itself onto the codon where the codes are. Okay, and the translation will begin with AUG. So A is definitely going to pair up. Yes.
Please, my humble suggestion, I wish that you use the cursor to point any place that you are talking about, because I just look at it. I don't know
Do you
when
see
you
it?
talk about.
You see the cursor?
Yes, sir.
You see it? Okay. So, like I was talking about earlier,
Hello,
the
sir.
code. Yes, please.
Hello, sir.
Hello, I'm listening.
Please, can you enlarge it a little because we can't see it well. If it can be enlarged a little for us. Okay, thanks.
Is it better now?
Yes, yes.
All right. Okay. So all that I was saying was that you realize that messenger RNA, there's a messenger RNA which has been mirror copied from the DNA. It is now in the cytoplasm. It has left the nucleus and it's now in the cytoplasm. And in the cytoplasm, there are lots of things, particles going on. You have lysosomes, you have Golgi apparatus, you have a lot of things. Endoplasmic reticulum, smooth, rough endoplasmic reticulum, all hovering in the cytoplasm. And the cytoplasm is a very watery substance. You have all these hovering in there. Then you have tRNA. The tRNA bears what? A set of anticodon. Whilst the mRNA bears the codon, it has codes, AUG, right? You see the codes that has been born here. Then you have this ribosome small subunit, the 30S. Then another large ribosome, which bears what? An amino cell site, a peptidal site, as well as what? An exit site. Is that right? Then the initiation factors act here. So the small ribosome would what? Attach or align itself with the messenger RNA. Okay, then this anticodon, the tRNA, will definitely find its way by pairing up with the codon. So you have an anticodon pairing up with what? A codon. And then initiation complex, that's mRNA plus the 30S. mRNA, okay? This is the mRNA molecule. Sorry, strand. This is the mRNA strand. Initiation complex, it begins here. So you have mRNA plus the 30S. And the 30S is this, right? This ribosome informs the 30S. Okay, there's subunits. Then the tRNA here will definitely align itself as a result of the formyl threonine enzyme. So translation begins with AUG on the codon. So AUG on the codon. Then you have the anticodon pairing up with the codon. So A will pair with what? U. Another A on the yellow strand is going to pair with the U on the messenger strand. Then the G on the messenger strand will pair with what? The C, the tRNA messenger strand. Okay, and that informs the initiation complex. It recruits the 50S. Okay, this 50 subunit ribosome would also attach itself to it, and it bears certain sites. That's the amino cell site. It informs NH2. Then the peptidal site, the proteinose site, also informs it. So the next tRNA in line moves into an A site. Okay, so the next tRNA, you realize that after it has formed, this one has formed, the next formation is going to occur in the amino cell site. So this tRNA was found hovering. So by the time that the initiation ends, there is another tRNA that has what? Been formed in addition. So that informs the initiation complex. Initiation complex. So first ribosome that occurs that there is another tRNA that has been formed in that direction. Now, let's move on to the next stage, which is the elongation phase of protein synthesis after the initiation stage. So the elongation stage of translation consists of 3 processes. 1, bringing each new aminosylated tRNA into line, and the aminosylated tRNA into line. That is the first stage. So this is the aminosylated tRNA. Okay, the aminosylated tRNA. You have an exit, you have a peptide, a peptidal site, and amino site. So the first thing on the elongation phase is that you bring each new aminosylated tRNA into line. Then forming the new peptide bond to elongate the polypeptide. So this is just a bit of what occurs. It replicates. So per the number of polypeptide chains that occur, and there are lots of ribosomes that will come attach themselves to the messenger RNA. It isn't just one. There will be lots of them occurring. So at the end of the day, you have a replicate of that. So it forms new peptide bonds to elongate the polypeptide. So what we just looked is just one aspect. So for poly, it means that there will be hundreds to thousands of it occurring, moving the ribosome along the mRNA so that the codons can be translated successively. So just as A would pair with U, G would pair with C, and it have to be 3 in triplicate, codon, anticodon, codon, anticodon, it pairs, and they'll be replicating themselves plenty in that regard. Now, let's look at a schematic diagram of the elongation process. A schematic diagram of the elongation process. So in the elongation phase, we still have the same diagram occurring, and methionine that has been transferred from its tRNA and forms peptide bond with the phenyl. Okay, so this occurs. This is what occurs in the initiation stage, right? It moves on into the exit stage and into the peptidal stage. And small subunits, it shifts. That's tRNA shifts to the E and P sites. The E is the exit site, and the P is the peptidal site, leaving an aminosyl site. Okay, so by the time there's what pre-location, you realize that what was in the exit stage will have left. Okay, it will leave the aminosyl stage. Then one would move large subunits shifts, new charged tRNA added to the A site. So one would move into the aminosyl site, whilst the one that initially was here in the exit site would leave into the cytoplasm. So schematic representation of the first stage of elongation of protein synthesis. So that means that it keeps recurring. It builds up, then one leaves and another picks it. It keeps on forming because there's not one amino, there's no particular type of amino acid forming. There are 20 different amino acids forming. So it depends on what type of amino acid is needed that has to be formed by the cell, by the existing cell. So that is what occurs in the elongation phase. That is what occurs in the elongation phase. Sorry. That is what occurs in the elongation phase. So elongation of protein synthesis. A key role is played by elongation factor EFTU, although a second protein of elongation factor is also required. So the eukaryotic counterpart of elongation phase TU is called elongation phase 1A. The elongation phase TU bound with guanosine triphosphate brings the next aminosylated tRNA into the A site. So you realize that initially it was in the A site and the P site. That was in the initiation stage. But when it came to the elongation stage, one had to move to the exit stage. And the exit will definitely move it into the open. So the EFTU bound with guanosine triphosphate brings the next aminosylated tRNA into the A site on the 50S subunit. Okay, which in this example is the tRNA F. This process requires the hydrolysis of the guanosine triphosphate to guanosine diphosphate. The 3' OH that we talked about earlier, that there was an attack on the 2' instead. So you realize that there is what? Hydrolysis of guanosine triphosphate to guanosine diphosphate. And once the GDP is formed, the EFTU has low affinity, the ribosome, and diffuses away. So that is why it is left. It moves out of the exit because it is not prepared to align itself to a codon until maybe it finds its own codon. It gathers enough energy to go about that bonding. So it has low affinity for the ribosome and it diffuses away, becoming available for reconversion. What? EFTUGTP, elongation phase TUGTP. So once the A site is filled, we have EPE on the ribosome, the large ribosome. It has 3 main sites: the exit, the peptidyl stage, and the aminosyl site, right? Once the A site, aminosyl site is filled, a peptidyl transferase activity catalyzes a concerted reaction in which the bond connecting the methionine to the transfer RNA methionine is transferred to the amino acid of the phenylalanine, forming the first peptide bond. So the peptidyl transferase activity is not due to a single molecule, but it requires several components of the 50S subunits, including several proteins and an RNA component called the 23S of the 50S. So some evidence indicates that the actual catalysis is carried out by the 23S RNA, which would suggest that 23 is an example of a ribozyme at work. So the entire cycle of charged tRNA addition, peptide bond formation, and translocation is elongation. So termination of protein synthesis. Are we going? What time do you want us to go on the break?
It's time.
Now.
Okay, all right. So let's end here and go for some 10-20 minutes. Then we return, right?
Sir, please be specific. 10, 20.
9:30. 9:30.
Thank you, sir.
Right? Yes. So 9:30, please. So welcome back. So we are on the last phase. We've looked at the initiation phase. We have looked at the elongation phase. Then we are now on the termination phase of protein synthesis. So the elongation steps out of our proteins were carried out repeatedly until a stop codon for termination is reached. So the stop codons are UA, UAG, and UG. No tRNA exists that can bind to a stop. So the tRNA holding the polypeptide, so it remains in the P site. But you realize we have three main sites: the E, that's the exit site; the P, as well as what, the A site. So specific release factors act to cleave. Cleave simply means to hold on the polypeptide chain from the tRNA to which it is attached, as well as what dissociates the 70S ribosome from the messenger RNA, after which the individual 30s and 50 subunits are recycled to initiate translation of another mRNA. 30 normally occurs in the prokaryotes, right? 30, 50s. The 50 are large ribosomes occurring normally in eukaryotes. So in here we have the termination stage. Let me enlarge it for you. So in the termination phase, you have a stopgap termination factor. That is, it goes into the A state, like then the exit. Okay, we have the exits, you have the P state. Then from the exit, you have the stop termination. The termination factor enters the A site, filled with termination factor. Then you realize that in the E site, there was a tRNA occurring in there. So that leaves the E site, leaving only the one in the P site. So the finished polypeptide is then released. Okay, finished polypeptide is then released. So there is a large subunit, that's a 50S, and the 30S is a small subunit. Okay, it's a small subunit. So you have ribosome cleared, subunit separate, and they are what, recycled. So these are the tRNAs which are formed, and there is a new one, polypeptide. Then a large subunit, there's a small one, and there's a stopgap. So you realize that it goes on and on and on and on, right? Because there are lots of what processes that occur. Initiation has to commence, elongation would definitely take place. Then finally, your termination. Okay, your termination. Okay. So the messenger RNA directs the synthesis of every protein molecule in a cell, originally copied from DNA. And protein production includes two kinds of processes. So these include the information transfer process, in which RNA-based sequence determines an amino acid sequence, and chemical processes in which amino acids are linked together. And finally, the factors that are required for translation to occur are the messenger RNA, the ribosomes, the transfer RNA or tRNA, and amino itself transfer RNA synthesis. That goes into initiation, elongation, and termination factors. So in summary, the process of translation is that an mRNA molecule binds to a ribosome, and the amino-sedated tRNAs are brought along sequentially, one by one, to the ribosome that is translating the mRNA. So just as we saw in the diagrams, because they are moving from the nucleus into the cytoplasm, there are lots of hot cell inclusions that are hovering in that watery ground substance in the cytoplasm. So it behoves on the messenger RNA or some ribosomes to align themselves onto the messenger RNA and undergo all those processes. So the amino-silated tRNAs are brought along sequentially one by one to the ribosome that is translating the mRNA molecule. And peptide bonds are made between successively aligned amino acids. So each time joining the amino group of the incoming amino acid to the carboxyl group, the amino acid at the growing end. And finally, the chemical bond that exists between the last tRNA and its attached amino acid is broken, and the completed polypeptide is removed. So realize that at a certain point in the termination, in the elongation stage, there comes a time whereby one of the tRNAs, which occupies the E site, leaves, and under normal circumstances, it's supposed to be a guanosine triphosphate, but when it leaves the E site, it becomes what? A guanosine diphosphate. So it has to undergo that form of synthesis to become what? An addition to become a guanosine triphosphate. Then again, it joins the large ribosome again and performs the coding sequencing to form protein synthesis. So that brings us to the end of this session, unit 4, session 4. We are now moving on to unit 5, session 5. And we are going to talk about the genetic code. The genetic code. Genetic code. So by the end of this session, you should be able to explain the genetic code and its properties. And again, understand and explain the mutations that may be associated with alterations in the nucleotide sequence. In the nucleotide sequence. So what is a genetic code? And per our earlier submissions, we have two nitrogenous bases, the purines and the pyridines, and they are made up of adenine, thymine, guanine, and cytosine. But thymine is replaced by uracil in the ribose or ribonucleic acids. But in the deoxyribonucleic acid, we have thymine as a nitrogenous base. So the four bases are adenine, thymine, guanine, and cytosine. They are sufficient to specify the 20 amino acids in proteins because each codon are 3 bases in length. Each codon is 3 bases in length. There's no way that you find a codon being 4 bases in length. They are always 3. Bearing a corresponding anticodon so that they are able to align themselves to each other. So each sequence of 3 adjacent bases in RNA is a codon that specifies a particular amino acid or chain termination. So the genetic code is a list of 4 codons and amino acid that each one codes. Each one has a specific code that it has to code. So before the genetic code was determined experimentally, it was written that if all codons were assumed to have the same number of bases, then each codon would have to contain at least 3 bases. So codons consisting of pairs of bases would be insufficient because 4 bases can form only 42, that's 16 pairs. 4 bases. So 3, 3, 3, 3, 3, 3. That would form what? 16 pairs. then triplets of bases would surface because 4 bases can form 43, which would be 64 triplets. 64 triplets. So in fact, the genetic code is a triple code, and all 64 possible codons carry information of some sort. So most amino acids are encoded by more than one codon. Most amino acids, they are encoded by more than one codon. So in translation of mRNA molecules, the codons do not overlap, but they are used sequentially. They do not overlap. They are used sequentially. That is why there are lots of codes that bears its corresponding anticodons. So genetic evidence for a triplet code, although theoretical considerations suggest that each codon must contain at least 3 letters. Codons having more than 3 letters could not be ruled out. The first widely accepted proof for a triplet code came from genetic experiments using R2 mutants of bacteriophage T4 that had been induced by replication in the presence of the chemical proflavin. So these experiments were carried out by Francis Crick and his collaborators. Proflavin-induced mutations typically resulted in total loss of function. So these were experiments that Crick conducted in the lab with some of his researchers, and the proflavin-induced mutations resulted in loss of function of most of the things that they researched into. So because proflavin is a large inner molecule, it was suspected that it caused mutations that inserted or deleted a base pair by interleaving between base pairs in the double helix. So instead of it performing other functions, it rather replaced certain base pairs or destroy them and actually try to bring up its own base pairs. So analysis of the properties of these mutations directly led to the deduction that the code is read 3 nucleotides at a time from a fixed point. So the research into it and found out that there's no way that the base pairs will be 2, but critical analysis revealed that they were in triplicates, they were in 3. In other words, there is a reading frame to each messenger RNA. So mutations that delete or add a base pair shift the reading frame and are called frame shift mutations. So the genetic analysis of the structure of the code began with an R2 mutation called FCO, which was arbitrarily designated plus R2 plus, as if it had an inserted base pair. So it could also be arbitrarily have been designated minus, as if it had a deleted base pair. So the designation of a positive simply means that there is an insertion positive. And when you deleted the base pair, you bring about a negative. So calling it positive was a lucky guess. However, because when FCO was sequenced, it did turn out to have a single base insertion. So the genetic analysis of the positive and minus mutations strongly supported the following conclusions. Translation of an mRNA starts from a fixed point. From the analysis of the mutations, it suggested that translation of mRNA starts from a fixed point. Then, again, there is a single reading frame maintained throughout the process of translation. Again, each codon consists of 3 nucleotides. So that brought the evidence for the fact that all genetic base parents were in triplicate and not in any other mode, but in three. Translation starts from a fixed point. Single reading frame maintained throughout the process of translation. Then each codon consists of what? 3 nucleotides. So elucidation of the base sequences of the codons. Peptide synthesis can be carried out in E. coli, just as we discussed. It's a bacterium cell nucleus. So extracts obtained by breaking cells open. So various components can be isolated. Various components of the E. coli can be isolated, and a functioning protein synthesizing system can be reconstituted by mixing ribosomes, because these don't have a true nuclei. It becomes easier working with them. But tRNA molecules, mRNA molecules, and various protein factors, if radioactive amino acids are added to the extract, then radioactive polypeptides are made. Synthesis continues for only a few minutes because mRNA is rapidly degraded by nucleases in the mixture. mRNA is rapidly degraded by nucleases in the mixture. So the elucidation of the genetic code began with observation that when the degradation of mRNA was allowed to go to completion and the synthetic polynucleotide polyuridylic acid, poly, was added to the mixture of an mRNA molecule, a polypeptide consisting of phenylalanine, Fe, was synthesized. Phenylalanine. So from this simple result, because there's an insertion, elucidation, there is an insertion. So from this simple result, the knowledge that the code is a triplet code, it was concluded that triple uracil, must be a codon for the amino acid phenylalanine. And variations on this basic experiment identified other codons. So a summary of the code, the in vitro translation experiment, which used components isolated from the bacterium, E. coli, have been repeated with components obtained from many species of bacteria, yeast, plants, and animals. So the standard genetic code deduced from these experiments is considered to be nearly universal, because the same codon assignments can be made for genes in almost all organisms that have been examined. However, some minor differences in codon assignments are found in certain protozoa and genetic codes of organelles. So here we go with the genetic code. Here we go with the genetic code. I believe you can see. Is it visible?
Yes, sir.
Yes, sir. It's visible.
All right.
Yes.
All right. So you realize with phenyl, you have UUC. Okay. With phenylalanine, and with leucine, you have UUA, UUG. That has to do with what? Leucine. And that is a uracil. Okay, uracil, and with a cytosine. The cytosine, you have CUU, CUC, CUA, CUG, right? Then with adenine, we have AUU, AUC, AUA, AUG. This has to do with a methionine, or a start or leader adenine. Then with guanine, we have GUU, GUC, GUA, GUG. That forms the valine. These are all type of amino acids. are all types of amino acids. So in the C, you have all C, UCU, UCC, UCA, UCG, the serine. Then proline, we have CCU, CCC, CCA, CCG. So all these are first base triplets or triplet codons occurring in the first and the third phosphate molecules, and the bases, the phosphate and the first base, you have the 5 end, the 5' and the 3' ends, and that informs the genetic code. So within it are these nitrogenous bases, which forms the codons, and out of this they form the various amino acids. You form the various amino acids, and these are codons that bears the various amino acids. That bears the various amino. We have 20 of them. So you could see 1, 2, 3, 4, 5 in the first row. 1, 2, 3, 4, and then you have 1, 2, 3, 4, 5, 6, 7. Then you have 1, 2, 3, 4, 5, 6. Right? You sum them up, and you are going to get your 20 different types of what? Amino acids. Amino acids. Altering the nucleotide sequence. So the codon assignments shown in the figure I just displayed completely consists with all chemical observations with the amino acid sequence of wild type and mutant proteins. So in virtually every case in which mutant protein affects by a single amino acid in the wild type form, so the amino acid substitution can be accounted for by single base change between the codons corresponding to the two different amino acids. You definitely have anticodons in that regard, which comes to play when the need arises. Well, for example, substitution of glutamic acid by valine, which occurs in sickle cell hemoglobin, results from a change from GAG. That is the normal. Code. But if it changes to GUG in codon 6 of the beta globin RNA, then it means that a sickling hemoglobin has what emerged. A sickling hemoglobin has emerged. So in contrast, a silent mutation is one that does not change the amino acid sequence, altering the nucleotide sequence. A silent mutation is one that does not change the amino acid. It maintains the same mother amino acid. So silent mutations often result from changes in the third codon position. For example, a mutation that changes an AAA codon into an AAG. So you see the third codon is what changes. It's silent because both codons specify lysine. Both, because they are same. So they overshadow the third one. But if it comes in between, then probably it wouldn't be a silent codon. A most interesting class of mutations consists of changes that convert a codon that specifies an amino acid into a chain terminating codon. So a mutation of this type is called a nonsense mutation, and it results in premature termination of the polypeptide chain. A nonsense mutation. It is premature. That means that it does not yield up to maturity. So before it matures, it perishes. An example of a nonsense mutation is found in the beta globin gene, in which a mutation from AAG to TAG in the 17th codon results in a truncated polypeptide and only 16 amino acids in length. So this mutation is one of the several types associated with the disease beta thalassemia. Beta thalassemia. So characteristics of the genetic code are given below. One, there is specificity. The genetic code is specific. A specific codon always code for the same amino acid. There is specificity. A would always pair with what? A U. There is specificity. A would always pair with a U. And with the same amino acid. G would always pair with what? C. G would always pair with a C. There's no way A is going to pair with a C. So there is specificity with regards to the genetic code. Universality, there is universality. The genetic code is universal. That is, the same codon is used in all living organisms, be it prokaryotes and eukaryotes. They all have same components: adenine, guanine, cytosine, thymine, or uracil, depending on be it ribose sugar or deoxyribose sugar. Then degeneracy. The genetic code is degenerate. That is, although each codon corresponds to a single amino acid, one amino acid may have more than one codon. Example, arginine has 6 different codons. They degenerate. Arginine has what? 6 different codons. So, in short, genetic code is the nucleotide base sequence on DNA and subsequently on mRNA by transcription, which will be translated into a sequence of amino acids of the protein to be synthesized. So the code is composed of codons. The codon is composed of 3 bases. Example, ACG, UAG, UCC, CCC, CAC, and each codon is translated into one amino acid. Each codon, that 3 code, is translated into one amino acid. And there are how many of them? There are 20 different amino acids. So all those 61 codons specify amino acids. 61 codons. 61 codons. A codon is made up of 3 triplicates, 3 nitrogenous bases. And in many cases, several codons direct the insertion of the same amino acid into a polypeptide. So this feature confirms the inference from the R22 frameshift mutations that the genetic code is redundant, degenerated. It is redundant, has gone on, let's say, a long break, or it has degenerated. So in a redundant genetic code, some amino acids are encoded by 2 or more different codons. In the actual genetic code, all amino acids except tryptophan and methionine are specified by more than one codon. The redundancy is not random. The redundancy is not random. Mutations that change the nucleotide sequence of a gene may differ in their consequences, and a special terminology is used to describe them. A missense mutation results in the replacement of one amino acid by another. For example, the change from GAG to GTG in DNA of the gene for beta globin results in the replacement of glutamic acid by valine in sickle cell hemoglobin. Then the mutation is therefore a missense mutation. So in contrast, a silent mutation is one that does not change the amino acid sequence. And they often result from changes in the 3rd codon position. As the example, triple A codon to an AAG codon silent because both codons specify IC. A most interesting class of mutations consist of changes that convert a codon that specifies an amino acid into a chain terminating codon. Then mutation of this type is called a nonsense mutation, and it results in premature termination of the polypeptide gene, as discussed earlier. So, an example of a nonsense mutation is found in the beta globin gene, in which a mutation from AAG to BAG in the 17th codon results in a truncated polypeptide, only 16 amino acids in length. And this mutation is one of the several types associated with the disease beta thalassemia. Beta thalassemia. So that is a summary of the proceeds in session 5. We are now moving on to the final session, which is session 6. Session number 6. Session number 6. Just a moment, please. All right, welcome back. In session 6, we'll be looking at the overall process of gene expression. The overall process of gene expression. And by the end of this session, students should be able to explain the processes of gene expression and understand the key events that characterize gene expression. What constitutes gene expression? So mechanisms of gene expressions are complex. Nonetheless, the basic process is a simple one. A base sequence in a DNA molecule is converted into complementary base sequence in an intermediate mRNA. DNA double helix structure unwinding to release one mirror copy strand. Then one serves as a template. Okay, and then the base sequence in the mRNA is converted into an amino acid of a polypeptide chain using tRNA molecules, each charged the correct amino acid. So both of these steps have a multitude of sub-steps, utilize the simplest of the principles. There are some principles that we'll be looking at. The rule of base pairing provides the base sequence of the mRNA. And earlier, we've acquainted ourselves with the base pairings. Adenine always pairs with what? Uracil, because we are dealing with what? Ribonucleic acid. Adenine always pairs with uracil, and guanine always pairs with cytosine. So the two-ended molecule, tRNA, with an amino acid attached at one end and able to base pair with RNA bases at the other, translate each set of 3 bases into one amino acid. So the sequence organization of a typical prokaryotic mRNA molecule is that the mRNA molecules are polycystronic, which means that they contain sequences specifying the synthesis of several proteins. A polycystronic mRNA molecule must therefore possess a series of start and stops codons for use in translation. Review of facts about gene expression. The flow of information from a gene to its product is from DNA to RNA to protein. So that is what occurs with regards to gene expression. There is a DNA that has to unwind, form an RNA, and through RNA formation, then we undergo the synthesization of what proteins. So the flow of information from a gene to its product. The gene is the DNA. That is the mother traits, genetic information. So it has to move from the DNA to RNA, then to other forms. That is a protein or amino acids. So the properties of the different protein products of genes are then determined by the sequence of amino acids of the polypeptide chain and by the way in which the chain is also folded. So each gene is usually responsible for the synthesis of a single polypeptide. Each gene is usually responsible for the synthesis of a single polypeptide. So DNA to mRNA to tRNA to aminosylated tRNA or ribosomal RNA, that informs what? Protein synthesis, giving up lots of what? Peptide bonds, polynucleotide peptide bonds occurring, polypeptide bonds occurring. So gene expression begins with the enzymatic synthesis of an RNA molecule. That is a copy of one strand of the DNA segment corresponding to the gene. So it is serving more sort like a repetition. Okay, there is a higher authority. Let's say the central bank. The central bank in every country is the mother bank that oversees to the day-to-day activities of all banking institutions in that country. So it is out of the central bank that you have the commercial banks, you have the various banks, banking institutions such as the rural banks, the savings and loans, and the cooperative unions, and they are all acting in that stead. So it boils down to the fact that the gene expression also begins with enzymatic synthesis of an RNA molecule. That is a copy of one strand of the DNA segment corresponding to the gene. So immediately it leaves the nucleus, the mirror copy, and the template is left behind. The messenger moves in there. There is an initiating phase. Then the ribosomes that are hovering in the cytoplasm then come and align themselves to their codons. Anticodons are also there. The transfer RNA is also there. We have ribosomes also hovering in there. And the ribosomes are made up of 3 sites: E site, the exit site, the peptide site, as well as the amino site. Okay. And out of it, they have their corresponding genes. So this process is called transcription and is carried out by the enzyme RNA polymerase. So this enzyme joins the ribonucleoside triphosphate by the same chemical reaction used in DNA synthesis. So RNA polymerase differs from DNA polymerase in that a primer is not needed to initiate the synthesis. In DNA, primers are needed to actually progress or start the processes. But in RNA, because they are made of just ribo, they are made up of nuclear materials. They are not really made up of nucleuses. They can just begin the process, unlike in DNA, whereby you have two nucleus and it has to go through a very well-structured stage or process. So DNA polymerase differs in that regard. So DNA would require a primer, like in an RNA, whereby there is no primer with regards to the enzymatic reaction of the RNA polymerase occurring to initiate the synthesis. So transcription, on the other hand, is initiated when RNA polymerase binds to a promoter sequence. Transcription is initiated when RNA polymerase binds to the promoter sequence. And each promoter consists of several subregions, of which we have two, of which two are the polymerase binding site and the polymerization start site. Now let's look at the polymerization. It continues until a termination site is what? Reached. Polymerization. It continues until a termination site is reached. So it begins from an initiation. It has to go through the elongation, then finally the termination. That is where polymerization ceases. So the product of transcription is what? An RNA molecule. RNA molecule. Transcription is simply DNA unwinding itself, leaving the mother template strand, one strand, leaving the nucleus into the cytoplasm, carrying a mirror copy of the information that has been left into the cytoplasm to begin the ribose sugar formation and protein synthesis. So, in review, as part of the gene expression mechanism, base sequence in a DNA molecule is converted into a complementary base sequence in an intermediate molecule. And then the base sequence in the mRNA is converted into an amino acid sequence of a polypeptide chain using tRNA molecules. And each is charged with the correct amino acid. Each is charged with the correct amino acid. Each codon is charged with a particular amino acid. So when the information in nucleic acid molecules is used, base sequences provide information for the first process. That is, specific sequences in the DNA are recognized as the beginning, that's promoter and end, transcription, termination type of a gene. These sequences are recognized by an enzyme, RNA polymerase, that makes a copy of the gene that is used by the protein synthesizing machinery. So to ensure the correct amino acid sequence is assembled, one codon, example AUG, is used to tell the system where to start reading. And a stop codon defines the end of the polypeptide chain. So particular recognition sites in tRNA enable the aminosyl tRNA synthase enzymes to connect amino acids to the tRNA molecules with the correct antigen. In certain groups, you might not come across amino acyl tRNA, but you come across ribosomal rRNA. rRNA. When you come across that, you shouldn't get confused. It is referring to the amino acyl tRNA because ribosomes are made up of certain proteinous end. You have the amino acyl as well as the peptide inside site before the exit site. So you shouldn't get confused. Then the key event of the entire process of gene expression is that both DNA and RNA are scanned by molecules that move in a single direction. So RNA polymerase therefore moves along the DNA as it polymerizes nucleotides. And the ribosomes and the mRNA move with respect to one another as different amino acids are brought in for covalent linking. So that note, I bring my lecture to a close. I bring my lecture to a close. And please, when am I? Yes.
Hello, sir.
Last week, I think section 5 and 6 we didn't go through.
Hello, sir.
Section 5 and 6. Yes. We were unable to finish section 5 and 6, right?
Yes, please.
So let me also put my own across, then you see the way forward. Thank you very much. And then on behalf of the class, I'll say, may God we wish you speedy recovery. I know you are not feeling well, but because you love us, you've taken the pain out and then be with us for over 3 to 4 hours now. We pray that all the energy that you've lost, may the Lord replenish it and give you speedy recovery to continue your day's activities. That is 1. 2, from the timetable and what we discussed the last time, this happens to be our last meeting, if all things being equal. So we'll be preparing for our quiz, end of semester, and then quiz 2. I want to put in a very humble suggestion, if you wouldn't mind. We are very happy. Oh, I think I have left. Hello, sir. Yes, sir.
I'm here.
Okay. We'll be very happy if you can give us a structure for our end of sem and our quizzes, if it will not worry you. And then the class will also be happy if we can look at it from this point. We use Unit 1, Session 1 to 6 for our Quiz 1, and then Unit 2 and Unit 3 for end of semester, and then the Unit 4 that we just finished for our Quiz 2. If we can structure it that way for us. Unit 1 has 6 sessions, if we can use all for our Quiz 1, and then Unit 2, Unit 3 for end of semester, where we have both subjective and objectives, and then Unit 4 for our Quiz 2. That will be the LMS 1, if you wouldn't mind our humble appeal, because it is quite voluminous. It is quite detailed.
It is voluminous. I heard you, right? And I will consult the coordinator. You see, at the nick of time, what actually happened was that at the nick of time, that was when I was consulted. Okay. And I'm not with UCC full time. I'm a full-time lecturer at the Department of Biology Education in Wilbur. So it was just about 1 week to the commencement of your start that I was giving this, right? And this is my first time of handling such a course. I used to do population genetics, evolution with code, and plant physiology. Those are the courses I was handling earlier. So it was dropped. I think I couldn't have time to handle those courses last semester, and so I was just consulted at the last minute to pick up this course. I don't have the ultimatum of what you are talking about, but I'll talk it over with the coordinator, Professor Foster P. Okay, I'll send your concerns to him, and they are in charge of the questions, blah blah blah. Mine is to just lecture, and they are in charge of that, and they have a bank, test item bank that they normally select their questions from. It's rather unfortunate that it has to end this way. That was why I was even of the view that even I might find some time to make it up so that you finish everything. Because it's not fair. It's not fair on the other side trying to, leave you this way at your feet. I don't know whether you've been given the models yet or not. I'm not informed about that.
And then the last request, sir, if you can make the model available, because we have
I don't have that power. I've told you. I don't have that power. I talked it over with the coordinator, and he was like, some people are supposed to. You are supposed to pay fees, and from that is when your models are given to you. But I don't understand why, as at now, you don't have it. It is beyond me. It is beyond me, but I'll still talk it over with the coordinator to see the way forward, because it's not in your best interest for this thing to occur to you. Is that right? So all that you have said, I've noted it, and I'll send your concerns to.
Kindly do that on my behalf. Thank you.
Hello, Mr. Jimai. Mr. Jimai.
Yes, please.
I don't know. I have the model. I downloaded it from our platform. I don't know if we could check back. It was dropped on our platform, and I printed a copy from it. So I think if we can consult, I think Castro dropped it. So you consult him to do that for us, to repost it for the usage of those who don't have it.
If you have it, I think that is it's a structured institution, and you don't do somebody's job, or else you are sent to court. Some of these things, that is how they work. So,
well noted. Second point, with respect to that,
you have raised a lot of second points. So.
Yes, that was the first one. The second point has to do with the submission of assignments. We don't have your details? Are we doing it through email or through your WhatsApp line? If we can make it available.
Yeah, so send it via my WhatsApp. Okay.
All right. So I believe you all have sales contacts.
No, please. No, please. If you can share it.
Okay, so, sir, if you can make it available.
And so just write it 0244 77 3097 0244 77 3097.
Okay, sir, what's the format? Word, PDF, font? Because usually they will know them. PDF.
Yes.
Okay, so police, if you've heard it, so PDF. Font 12.
Times New Roman 12.
Thank you.
The number again, please. I want to confirm.
0244 77 30 97.
Thanks.
And no, you don't call. You don't call. Nobody should call it. I have a whole community under my jurisdiction, so you don't call. Don't waste your time calling. Just send your assignments. I'll attend to it.
So please don't forget to add your index number, your name. These are the
Course
things that
title,
course title,
course code.
Course code, any possible the lecturer in charge.
Is
Dates as
deadline?
well. Deadline Wednesday.
Wednesday. Hey, wow.
Wow.
Who said wow?
Yeah, the two. All the two on Wednesday.
Yes,
All the two on Wednesday.
Hey,
Hey, wow.
They are
yes.
related.
Yeah, same one.
They are related. So if you have a question,
Hello,
Nobody
sir.
told you. Yes, nobody told you to come
you
to
are
school. You said you
Oh, sir,
come to
please,
school.
please extend it for us, okay? Please.
I don't have the time, please. I don't have the time. Listen,
Please, Friday
I told
will be
you
best.
it will be best for you, not for me. I'm a chief. You get it? And I have lots of responsibilities on me. I'm not feeling well, but I have to, wake up and prepare for this lesson. So your convenience is not my convenience, please. Wednesday final. I can't go beyond that. And I'm going on a long break. I need a long break before I break down.
Okay, Nana.
So on that note, I wish you all the best and stay blessed.
Mr. Adman.
Julie, also friend, why?
Effie Juliana. Juliet. Aye. Julie. Juliet. Okoda. Aye. Monko.